I'm interested in fungal biology and population genetics, and my background really is in mating systems in mushrooms, basidiomycetes, specifically Armillaria. But a few years ago, we had sort of an epiphany, and that was that we could do experiments to ask evolutionary questions. And to me, I'm at heart an experimentalist, so this was a really good fit. And then a few years hence, Jeremy Detman, who was a student with John Taylor at the time finishing, and Linda Cohn in Toronto and myself, we unhatched a plot to use Saccharomyces to ask even more general questions. So we're interested in the process of adaptation in natural populations and the underlying determinants that go along with that. But we realized we could address a more major question. And so that's what we started to do when Jeremy came on the scene as a postdoc in Toronto. So the main organism today is Saccharomyces cerevisiae, but it actually turns back to some of my older work on an older system, Armillaria, toward the end. So think of that as a short appendix on the main part of this talk. So this is the organism, Saccharomyces cerevisiae. And here is the question. Okay, we became interested in divergent adaptation and its possible role in speciation. And we realized that we could use yeast to study this in a unique way that had never been done before. And so here are the bare bones of the system. Imagine a population going along in time. It's got mutation. It's got genetic drift. It has natural selection. We can allow genetic exchange and recombination to happen or not at will in these experimental populations, but this is how it's going to happen in nature. So mutation, there's a certain amount of variability in the population. There may be a geographic barrier to mating, so populations become subdivided. That could be continental drift, or it could be the experimenter using a pipettement to subdivide one liquid culture into many. And now these subpopulations find themselves in different environments where the features of selection work in different ways. So the same population genetic pressures are going along, and different alleles and different genotypes are favored by natural selection. There may also be a random component due to genetic drift, but overall fitness goes up in these populations. They adapt to their respective environments. And so this is divergent adaptation. The two populations adapt in their respective environments. They're not yet species, and that's an important point. This is what we expect to happen under the allopatric model initially. So this is incipient speciation. These are not yet species, but the prediction is that should they hybridize, fitness goes down in those hybrids. And there are two components to that fitness decrease that we can quantify in yeast that are not so easy to quantify in any other kind of experimental system. So the first reason for the decrease in fitness is ecological isolation. And that's simply because the hybrids are an intermediate genotype that are no longer optimally tuned to the respective environments, and so fitness goes down. But there's another component, and that may be that certain alleles found in the different respective environments, when put together in the same background, may clash, and the downstream effect on fitness may be negative. So these are not mutually exclusive. We've got one and the other, and with yeast we can quantify the contributions of these two modes of reproductive isolation. So not yet new species, but we do predict reproductive isolation. So here's the take with yeast. We realized we could recapitulate this whole process starting with a single genotype, a single fungal cell. And through evolution, we could observe all of these processes happening. So that's probably the most important take-home lesson of the whole talk, is that all the diversity in genotypes and phenotypes come to a most recent common ancestor in a single fungal cell. And the immediate descendants of that are frozen in the -80 freezer. So that was the idea. And the experimental system is simple. There's our single ancestor, which has grown up into a colony. We start liquid cultures. We use batch culture method rather than chemostats. So there's a daily boom and bust cycle, but an average number of generations. And then on the practical side, all of our strains are barcoded. And I remember making these barcodes by tossing coins, 2 coin tosses for each base position, and simply read that out, and that's where they are. They're part of the strains now. We use those in a number of ways. It's a check. They represent a check on contamination, which can easily happen in these experimental systems where there's a daily probability of a mistake, and you need to be able to identify that when and if it happens. And we can also use the barcodes to quantify the abundance of a particular genotype in a mixture of cultures. And we're using a very simple measure of fitness. It's just basically the number of doublings. But we use barcode abundance to get at this quantity. Okay? This is a simple measure of fitness. It's much more complicated in nature, but it works for our immediate predictions. And also with next-gen sequencing, the first thing we do with a sample is recover our barcodes right off the bat to make sure no one has messed up along the way. So it's a simple experimental system. And then here's the experimental design. We've got the single ancestor, the progenitor. We had 5 populations in an environment high salt. We selected that because a lot is known about salt response in yeast, and also low glucose. And so the progenitor is divided up, barcoded. We have 6 of these populations, 6 of these populations, and we did 500 generations of evolution. This is the Detman et al. Paper. It's now ancient history, but we've been mining out these populations ever since in terms of the underlying determinants of what happened. So we study these populations in sets. And here's how that happens. Start with a progenitor. We evolve a subset in high salt, low glucose, M, and now we can make the hybrids between those two. So this is an SM hybrid, and these are made at the population level. We select out all of the unhybridized genotypes to get those. But we can also make another kind of hybrid, and that's the salt evolved with the progenitor, SP. And if we measure the fitness of these 2 kinds in high salt, notice that they differ from one another. This one will give us the measure of ecological isolation because we've essentially diluted the S determinants in half. This one, we've done the same thing. We've diluted the S determinants in half, but now we have M determinants. So this one should have lower fitness even than that. And that's going to allow us to partition the quantity of effect of ecological versus Dobzhansky-Mueller genic incompatibilities. And we can do the same thing on the low glucose side. So we had 6 sets of these, and we always saw the same effect. Fitness was always lower here than here, and this usually was lower than this. So in other words, we've got ecological and DM incompatibilities going on. And here are the data in aggregate. Don't have enough time to present the individual ones, but you see the different kinds of hybrids here on the x-axis. This is relative fitness, which we measured in competition experiments using the barcodes as a measure of abundance over time. This line equals the fitness of the ancestor. And here, this is just the permissive environment, rich medium, no significant effects of evolution in either the pure high salt or low glucose evolved or any of the hybrids. But look at the high salt environment. This is the salt evolved, huge increase in fitness. That's a giant fitness increment. And look at the SP hybrid. That's going to give us our measure of ecological isolation. And the SM hybrid, it's got the same amount of high salt determinants, but it also has a contribution of M genes. That's the negative interaction or negative epistasis. something is present in S and M that is interacting in a negative way. So we're trying to get at what is the underlying nature of that interaction. So those are the old data. And what are the underlying determinants? So we did some next-gen sequencing. We took isolates initially as from the populations, single colony isolates. So we're measuring individual genotypes within the populations. 119 of those distributed over the 12 subpopulations, 6S and 6M, sequenced to about 100x depth. And the disheartening thing for me as a non-bioinformatician in the beginning was the return on the SNP calls was huge. There are just too many SNPs in those tables. What is all that? An average of about 700, where we're thinking back of the envelope calculations over time, there can't be that many mutations that are coming along with single colony isolates. Collectively, in the population as a whole, there are hundreds, thousands of mutations, enough to cover the whole genome. Every base position will be mutated on average, but they're all rare. So when you take a single individual, we shouldn't be recovering that much variability. On average, we were. But there were several sources for all of that. And first is our ancestor differs from the sequenced reference by about 500 SNPs. And they're easy to identify because they occur everywhere across all of the populations. We just filter those right out. There are alignment and sequencing artifacts, systematic errors. And this is curious because if you specified, say, 100 sites in the genome at random and gave me those coordinates, NextGen can achieve 100% most of the time in terms of making the correct call. But in every sequencing run, there's a tail of anomalies, and over a large genome, that's a lot of stuff that needs to be sorted through. So we need to remove those. And for the purposes of this talk, I took out the singletons and doubletons. So the mutations of most interest are those that are found in different isolates of the same population, but nowhere else. And that indicates we're coming into them repeatedly. Perhaps they're important. Let's give them more priority right now. And I'm even taking out the doubletons. So what do the data look like? So here we have the high salt populations. And in this graph, these are the mutations, the locations and coordinates, nature of the change. Home stands for homozygous. So the mutation is homozygous in this isolate for that mutation. Het means heterozygous. And blank means was not recovered in that particular isolate. So the first thing you notice is that we've got a lot of named genes here. And within that, most of the changes are predicted to be amino acid substituting changes. The named part is not that surprising. 6,000 genes, 10,000 labs worldwide. There's a lot of background. So most everything is known. And there aren't many non-coding mutations in this list. This one is an exception. There's no name. It's a non-synonymous mutation, but it has a signature that suggests that it's important. We have 2 representatives here, and then it's much more common at the 500 generation time point. So there's a signature there that suggests we should not ignore that one. And then look at all the PMA1 mutations. In fact, there's another PMA1 down here that didn't make our filters, but we know it's real. By the way, we've got verification of all these with conventional PCR and Sanger sequencing. So PMA1 must be important in high salt. And I have to warn you, there's a quiz coming on this, okay? Because PMA1 is an ATP-driven proton efflux pump. And it's important for that proton gradient across the cell membrane. It drives a lot of physiological processes. What does that have to do with resistance to high salt or tolerance to high salt? So there's the pre-question for the quiz to get you thinking about it. All those PMA1 mutations, it's a feature of the high salt medium. Other examples like this one, that's a leucine aminopeptidase. I have no idea what that has to do with high salt. There's a LAP2 polymorphism in mussels that's associated with living in different salinity of environments. I don't know. Could be important. We don't really know. There's another gene, TUP1, that's mutated, and its partner, Psych8, has been mutated. These are global repressors, and one of the targets of TUP1 is a group of genes I'm going to tell you about called the ENA genes. They're involved in sodium efflux. And so what this is loss of function in a repressor for a gene that's involved in sodium efflux. So this is going to be a recurring theme that can come up. So low glucose, now it gets a little more complicated. The phenotypes are a lot more involved because we need to think about the growth mode of Saccharomyces, and that is that this organism favors fermentation initially, whether oxygen is present or not. If it's got glucose, it's going to ferment. And then after a period of time, when the glucose is exhausted, the fungus has produced ethanol, it'll switch to respiration. The cellular wiring will change extensively at that time, and it's called the dioxic shift. And it may be a kind of evolutionary strategy. Use the glucose inefficiently, poison the competition with ethanol. When that's all done, reuse the ethanol in respiration. It's a just so story, but it's an attractive one. Okay, so that may be happening. This initial effect, it's known as the Crabtree effect, and the dioxic shift is where metabolism changes. So what you get is the early mid-exponential phase, and then the dioxic shift, and a long respiratory phase. This is a low glucose evolved strain. That's the progenitor. And what we find is there are mutations that affect this phase and other mutations that affect that phase. So we have to keep those in mind. So here are the low glucose populations, M7 through 12, same layout. A couple of things to note here. Look at all the mutations in RAS1 and RAS2. That has to do with sensing and control of cell division. We don't know exactly how, but it's a recurring theme. But there's a very powerful program of adaptation going on here. MDS3 is a gene that affects the fermentative part of the growth curve. And MKT1 has a major effect on the respirative phase. And together they work synergistically. And we don't know what this one is doing. But both of these genes are coming up to near fixation at generation 500. So that's going to be an important thing to keep in mind. So let's just focus on two populations. And this is a more detailed study of frequencies over time. So here what I'm doing is not isolating effectively single cells, but taking a mass isolate from the population at different time points and then sequencing to about 1,000x depth and keeping track of the various mutant alleles. So you can see here, look at all those PMA1s. There's not just one, there are several. And they're coming up to different frequencies by generation 500. There's our repressor, it's coming up. LAP2 is there. This one is actually a methionine auxotroph, but it seems to have an interaction with some other genes, and there's a reason why this has increased in frequency. We don't quite understand it. But you can get the idea there's a lot of competition among genotypes. And that competition is called, what do you call that? When you have a bunch of genotypes competing with one another in effectively a clonal population. Pierre? Yeah, it's clonal interference. So there's a lot of that going on here. What about the ENA genes? These are the ENA genes occur in a cluster of 3 genes, but what's happening is the cluster is expanding over time. So this is Illumina reads mapped, proportion of Illumina reads that map to ENA over the whole chromosome, and you can see it's going through the roof. So the copy number of ENA genes is going way up. So what the populations are doing is overexpressing sodium efflux pump genes. They're doing that by duplicating the cluster, and they're de-repressing the cluster at the same time. So that's a predominant theme here. M8 is a much simpler story. Here we have MDS3 going up first in frequency, then MKT1, and then this third one we don't understand. This has to go in this sequence. We've tried to evolve this one using that as a background. It doesn't work. We can evolve this one if we use that as a background. So this sequence has to happen in that order. And so what do we see in terms of patterns? Two very different patterns. We've got clonal interference in the high salt situation. M8, we have a rare but very powerful program of adaptation. Only 1 out of the 6 low glucose populations, but it's important. Not much evidence for genetic hitchhiking, and this differs from another paper that was published recently, which showed a lot of genetic hitchhiking, but under somewhat different conditions. So those are the situations. And now, okay, we have the determinants, but we felt it's sort of incumbent on us to learn about how those determinants work. So here are the ones we're looking at. We've got in high salt, PMA1 is obviously important. We have the sodium efflux genes. These are both ATP-driven proteins. Over here, MDS3, it's a global regulator of transcription. This one is a global regulator, but not of transcription, it's of mRNA half-life. So it has to do with sequestration. And the genes affected are those that contribute to mitochondrial function. So this one acts early. It improves the fermentative phase, and then that improves the respiratory phase. And there's a major DM incompatibility between these two mutant alleles, PMA1 and MKT1, major negative interaction. So now back to the quiz. Why would mutants in PMA1 have anything to do with sodium efflux? Okay, so they're both ATP driven. This is about proton efflux, this is about sodium efflux. What might the mutants be doing to PMA1 that are going to increase sodium efflux? That's the quiz question. Anybody have an idea? Louise? No. I'm going to add. Okay, so what if you cut back on proton efflux? What does that do to sodium efflux? It's going to help it because they're both positive ions. So to some extent, it's a zero-sum game. And the hypothesis was that the mutants are actually causing proton efflux to lessen, and that enables more sodium efflux. Now this can have negative effects. Some of the PMA1 mutant cells are sick. They don't look right. But let me give you the evidence for this. So this is the scheme that I'm going to go through. And from here on out, all of the experiments involve strains that differ by defined nucleotide sites only in these genes, nowhere else in the genome. So everything else is homogenized. We're pretty sure of that because we bred each of these mutations to homogeneity and separated them from all of the other alleles. So we can put all of these back together in different combinations where we're just comparing single nucleotide sites and nothing else. So let's look at PMA1. Here's one of the mutants. What we do here is we take a cell population and starve it, and then we add back glucose and we look at the drop in pH in a lightly buffered medium. So here's the ancestor with no glucose, and you can see that even without glucose, it does acidify the medium. This is time, that's pH. Okay, here's the ancestor with glucose added, and you can see it acidifies the medium strongly. You can see an initial drop here when you add the glucose. Do you know what that's about? Why that initial drop here? It's too fast for any effect on the pumps, but you see a slight drop in pH. Well, glucose is acidic itself, so it actually acidifies the medium to some extent. MKT1 doesn't have much effect on acidification, but the PMA1 mutation slows it down. So this one's acidifying more slowly than the ancestor. So that was at least consistent with their idea that the mutants are causing a reduction in proton efflux, and that is going to enable more sodium efflux. And that double mutant does even worse than this one, because as you'll see, the evolved or the mutant alleles for these two genes, they hate one another. Bad things happen when they get together in the same genotype. So in terms of rates, the mutant allele here is acidifying much more slowly than the ancestral allele. And the internal pH is different. We did this with fluorescein dye analogs and flow cytometry. Some of the dyes are pH sensitive and others insensitive. And so you can get a measure of internal cellular pH. And for this mutant, the cellular pH is more than a half a unit lower than the ancestral. That's huge. That has a big effect on cellular physiology. So that's PMA1. Let's talk about the low glucose side. So this is MKT1, the mutant allele. This is the MDS3 global regulator. What you can see is when this one is used as a background and together with this mutant allele, it rises very quickly in the population. When the ancestral allele is used as a background, much more slowly. So the benefit of this in the respiratory side of the equation works much better with MDS3 working early in the growth curve. And so we tried to recapitulate this. And so we have replicate populations where we use the progenitor as a background, and we're going to look for these changes de novo. And we here saw no changes. Here we predicted that even starting with the ancestral allele of MKT1, that given the spontaneous nature of mutation, the large population size, we're going to see exactly that same mutation again appearing and being favored by natural selection. We think we know enough about the environment to predict evolution at one particular nucleotide solution. And in 2 out of the 3 replicates, that's what we saw. So here's one. So again, MDS3e is the background. This is the rise in the mutant allele. The ancestral allele goes down concomitantly. And there's the signal across the x-axis. So this happened de novo. Reviewers like to say, no, that's a contaminant. No, it's not a contaminant. It has the same barcode, and it is a mutant allele representing mutation within this time course, somewhere back here before this generation 87, where it started. So this is an aspartate to glycine change. We also had an aspartate to alanine change, practically the same thing. So this is the second replicate where the prediction was borne out, and not in the third. But that shows you can recapitulate this change. And why is it important? We think that there is an effect on the glucose transporters. So here, this is expression, see this, of transporter genes. And we think these first 4 are the most important ones. So this is the evolved allele of MKT1, which is always low, MDS3, which is usually high. There's the double mutant. And what happens is that even in the double mutant, expression is greater or at the median expression level. So in other words, the presence of the MDS3 mutation is helping the process of glucose transport early, and that ultimately has an effect on the behavior of the MKTe allele. They do better in combination in terms of growth than either does separately. And here's the DM incompatibility. So that's ancestral for both PMA1 and MKT1. So now we're looking at the negative interaction between PMA1 and MKT. There's MKT, mutant allele, PMA1 mutant, double mutant, and you can see the culture density is much lower. That represents a DM incompatibility. We still don't know exactly how that works, but it's not an effect at the level of the transcriptome. PMA1E has remarkably little effect on transcriptional patterns. In fact, none that we can detect, remarkably. MKT has a huge effect on transcription. So the interaction here is something other than a global interaction at the level of the transcriptome. But the interaction between these two does involve interaction at the level of the transcriptome. 163 genes where, plus those 4 glucose transporters, where there is measurable interaction occurring. And again, the MKT1 module here, this is the one that's important in the respirative process. So we don't know the details, but there is something very positive happening between these two. By the way, this allele here is one that's fixed in nature in Saccharomyces cerevisiae, and also in the closely related Saccharomyces paradoxus. We think that in labs, we always grow Saccharomyces, short growth periods, mid-log phase. We're emphasizing the fermentative phase only. The respirative phase is usually somewhat ignored. So our experiments are actually recapitulating the allele that is found in nature anyway. And how is this all put together? So these are various genotypes: PMA1, MDS3, MDS3, MKT1, MKT1 alone. There's the combination of mutant alleles that hate one another. We're looking at the budding index here, proportion of cells that are undergoing budding. And it's lowest here. It's not significant. But if you look at these colonies, they just don't look right. So essentially, we think that reduced expression of the transporter genes caused by MKT1 is actually exacerbated by the low pH conditions. That's the hypothesis. And at this point, I think I'm going to have to turn that over to the real molecular biologists, people like Louise and others who can test that greater. There is an overall effect. What have we got so far? We have divergent adaptation. The mutant alleles that we see have big effects on fitness. They're mostly global regulators, either regulators at the level of physiology, at the level of transcription, or RNA half-life, essentially. But they are global regulators. Certainly, there could be many more genes evolved. Some of those interact negatively. Well, that shouldn't be a big surprise because global regulators make big changes, and it's highly likely that there will be a clash. Not necessarily so. It doesn't have to happen that way. The interaction could be positive, but whenever you go making a big change and you put two of them together, bad things can happen. You worry about this every time you load a new piece of software on your computer. How is this going to work out? Usually it's not good. Okay, that can happen. So I think this is a good system to study evolution. If I had a phenotype or an environment of interest, I think people should do experimental evolution and compile a compendium of pathways involved, genes affected. And I think the big benefit here is that we're looking at alleles that have been already subjected to natural selection. And so I think this can complement functional interpretations. A knockout or a knockdown is one thing, but it's a rather blunt instrument. And I think that having a compendium of mutations, each with known effects, can really help our analysis as we go forward. But at this point, there's still a problem. I have a colleague at home who's always on my case. And he always says, Jim, what does this have to do with reality? These are little test tubes. I mean, this is a fantasy. And I say, yeah, I know, we've talked about that. And I always argue that the test tube environment is inescapably part of nature. I mean, it is. But I do worry about what he's saying, and I think there's a lot of legitimacy to it. How general is it? Because, you know, by the time we get to a population, it's been massively filtered by natural selection. We're not close to the mutations themselves, even in these experimental setups. We don't know much about the spatial location or the timing, or even the original spectrum of mutations. And so I thought, there must be a better way to look at this in general. So I want to just give you an appendix now, another little foray into this. How general can we look at the process of divergent evolution? And here the experiment was started before people knew that microorganisms existed. Okay? So that's the start of the experiment. And we're going to look at the endpoint now and hope to be able to get closer to the mutational spectrum. And that's the idea. So for that, I'm going back to Armillaria, the big fungus. Okay, so this is the honey mushroom. It grows on dead and decaying wood, and it makes these fascinating cord-like structures called rhizomorphs that radiate out into the soil and cause new infections. So the fungus has a tremendous capacity for vegetative expansion, and it can occupy many adjacent tree root systems over time on a forest floor. It's nearly ubiquitous. If you put in a woody substrate or a potato, very often they're colonized by this fungus. You see the rhizomorphs here. So I wondered if individuals of this fungus could be a way to capture the mutational dynamic. Now here, the experiment was set up a long time ago, not by people, and the selective conditions we can't do anything about. It's probably a matter of purifying selection. These grow in the humus, soil, probably minimal effect by climate change or whatever else you might want to think about. But at least we can study the process of divergence closer to the act itself. And so here's the way this is going to work. Each individual starts in a unique mating event. So two gametes get together, establish a diploid, which grows vegetatively over time. And the idea is that mutant sectors may be left behind in space. In other words, a kind of spatial record of the mutation dynamic, if this is true. And really, this is early stage, just proof of principle, if anything. So I selected an area for study where I had done collections 25 years ago, and I knew that there were two genetic individuals of this fungus on this island. It's a mixed hardwood forest. And sure enough, both individuals are still there today. We do genotyping using some sequencing of random DNA regions and also somatic compatibility reactions. There's a fascinating thing that happens when you pair individuals or isolates from the same individual in culture. They merge seamlessly. But when you pair isolates from different individuals, there's kind of a brown reaction line, probably a lot of cell death, I don't know. But it's a recognizable reaction. Think of it as sort of a histocompatibility response between different individuals. So they're genotyped, and we have two individuals. Call them individual 1 and individual 2. And we have a reference sequence for this individual. And it's a big genome for a fungus, about 86 megabases, low repetitive, high gene content. It's not a beautiful assembly, but it's certainly good for navigating the genome and tracking variability, because you can go to specific sites and amplify them reliably and all of that good stuff. And there were two collections that I resequenced and then compared back to the references. So I'm looking for sites that are heterozygous in one isolate and not in the other. And the problem here was just like my initial foray into Saccharomyces, too many candidates. In one case, comparing this to this, there were about 80,000 candidates, and this to this, there were about 60,000 candidates. That's too many. And when you look at those candidates randomly, just taking a random selection, most of it's false. Either the SNP is not there in any of the isolates, or it's there in all of them and it was failed to be called in one or more of them. So most of them are false. And I thought, that's not looking good right now. But if you apply some quality filters, and if you demand that when an isolate is homozygous, that there are no reads for the alternative allele, the candidate list comes right down to something on the order of 100, so that's manageable. And from among all that, the efficiency of finding the true variation is still low, but we found some. And it looks like this. So what I'm going to show you now is an exploded map. And it's a spatial map, but it's also a genotype network, okay? Because this individual's growing asexually, and these changes should occur in a phylogenetically consistent pattern, and they do. So first of all, there's one subsector that has lost heterozygosity. LOH means loss of heterozygosity in the rDNA cluster. And this has to be a loss event. It can't be a gain event because this indel is polymorphic in the population at large. So loss of heterozygosity. And then perfectly nested within that is another change with another LOH affecting 34 sites on the same chromosome segment over about 5 kb, and that's a gene conversion tract. That's what it's got to be. Plus another site has mutated, so the ancestor is homozygous, heterozygous, and within that, a subsector defined by 2 mutations, and then this one, 3 and 1, and then up here, another pattern, so that this is distinguished by 3 mutant sites from the progenitor, and that one. And so that's the spatial distribution of the little bit of variability that we found over the 86 megabase genome. There shouldn't be a lot of variability. What can we do with this? A couple of things. We can, it can tell us something about the development of the fungus as an entity, how it grew and how it colonized and what the spatial record is. This is only one individual, so we don't know whether the pattern is universal. But I think it can get us closer to the actual mutational spectrum under natural conditions. Of all the mutations here, 2/3 of them were non-coding. Okay, so that's quite unlike the yeast work, where almost everything was coding and non-synonymous, obviously important. This is probably more like neutral. 4 of them were in coding regions and a mix of synonymous and non-synonymous. So not enough for statistics, but it looks sort of neutral. The only mutations that we should be missing really are those that are both highly deleterious and dominant. So everything else should be preserved in the record. Think of this as kind of a mutation accumulation line, but in 2 dimensions with a spatial record. So there's the proof of principle. There's where it stands. And I better stop here. Thank you very much for your attention. Yeah. Thank you.